Okay, so what I wanted to do, right, is go over what macromolecules refers to. All life on Earth, right, use 4 macromolecules, right? The carbohydrates, the lipids, proteins, and nucleic acids, right? They are, and if you know those, that's a good thing. You know what they are, what they refer to. Now, lipids aren't necessarily polymers, right? As I said, they're a category of molecules that are hydrophobic. Okay. Now, the other 3, carbohydrates, proteins, nucleic acids, are polymers, which by definition, many monomers. Okay. And so I would expect you to know, like I might ask, what's the monomer for carbohydrates? And for at least the examples I've given you, like starch, glucose is the monomer, right? If it's lipids, there's no specific monomer, right? Fats are in this category, but you don't put, a monomer together and make a long chain, right? You put, in starch, you put about 1,000 glucoses, you link them all together to make one starch molecule. And then, say if you eat an apple, there might be billions and billions of starch molecules in the apple. So when you make a protein, the monomer for protein, there are 20 different amino acids. And then the monomer for nucleic acids are nucleotides. And your nucleic acids include, right, DNA and RNA. Okay. So, if I want to make carbohydrates, it's like starch, right? The examples are like starch, cellulose, and glycogen, right? These all require specific enzymes, and the enzymes would require glucose. So if, a plant, right, I'm not sure, it probably depends on the plant, what enzyme they use to make starch. I don't know, there might be 1 or 2 versions of it, I'm not sure. They would need plenty of glucose. They'd also need an energy source, right? When you link these together, right, the polymers are connected by covalent bonds, right? That's what holds these macromolecules. Lipids are held together by covalent bonds. The amino acids are, and the nucleotides are. They're literally polymerized. Right? Polymerized is a verb. Polymerization results in polymers. Now, the easy one is glucose, or carbohydrates in these, because right, glucose is a simple, it's just one, and it's many glucoses. Right? When it comes to like proteins, though, there are 20 different amino acids that you have to link together using a ribosome to make a protein. Right? There are, 8 nucleotides, really. You have 4 that make DNA, 4 that make RNA. Right? So if you want to make DNA, you use the nucleotide triphosphates with a little d in front of it. If you want to make RNA, you just use the nucleotide triphosphates. Right? So little d ATP, little d CTP, little d GTP, little d TTP, right? And then these, you have just ATP, CTP, GTP, and UTP. Right, so these four nucleotides are used by DNA polymerase to make DNA. These four nucleotides, right, are used by RNA polymerase to make RNA. Now you also make ATP, this one here, this version during cell respiration, and that's used for all sorts of things, like contracting the muscle. Right, so these are generally just used to make DNA, but these four nucleotides are not just used for RNA, they are used to provide energy for whatever needs energy. Chemical reactions, transport, turning enzymes on and off. You end up phosphorylating sometimes things with these as well. Okay, so the macromolecules refer to, at least the three of them are polymers, right? And these are, some of the examples of carbohydrates. Glucose by itself is also considered a carbohydrate, so it's a little bit different than the others. But like fats, phospholipid is another type of lipid, and then the steroids is another example of a lipid, right? And then proteins, when proteins are complicated because they do so many different things, so your hair is made of protein, you've got collagen in your skin that kind of helps keep your skin together. You got your tendons and ligaments are made of protein, so they kind of, keep your bones together. And then DNA and RNA, right? So when you sequence DNA, you say the sequence of DNA is A, C, G, or T, and that's the idea behind that, but so I just like to focus on the covalent bond. The other thing that there are two types of covalent bonds, right? If you make these, right, the bonds between the glucoses are considered glycosidic bonds. They're glycosidic covalent bonds. When you make proteins and you put the amino acids together, there's a peptide bond between the amino acids that's also considered covalent. When you make protein, you're going to have primary, right? There's the primary protein structure, and then there's secondary next, and then you have tertiary. I'm not sure that's tertiary, which is there's no structure with primary. You start to get it folded with secondary, and then tertiary, it's all put together at that point, right? The proteins form their conformation. Conformation is another way of saying the protein is folded properly. As long as it's folded properly, then the protein can do what it needs to do, right? Whatever job it has. It could be an enzyme, transporter, receptor, all kinds of, there's 8 of them altogether. I don't really focus on all the, biology class, we learn a little bit about, some of them, but not all. Okay.